Hello, I'm Dr. Stephen Pipe from the University of Michigan, and I'll be presenting at this ASH meeting a poster on behalf of my colleagues from Switzerland and Germany. I'll be taking you through the preliminary results of a first-in-human gene therapy study of adeno-associated virus capsid A237. This is a AAV technology that was selected to have broad patient eligibility and also hopefully produce stable and sustained long-term factor VIII expression in severe hemophilia. This is called BA2599023, and I'll call it BA259 from this point forward. This encodes a B-domain deleted factor VIII. The HU37 viral vector was selected because of preclinical data which had shown efficient liver-specific factor VIII gene transfer and favorable biodistribution, as well as durable factor VIII expression in those studies. The key aspect of this particular capsid, though, is that patient eligibility in almost all AAV-mediated liver-directed gene therapy trials for hemophilia has been restricted by pre-existing humoral immunity against the AAV capsids. So the main consideration while developing BA2599 was to select a capsid that would have low anti-AAV antibody seroprevalence. What we're reporting here is the preliminary long-term safety and factor VIII activity following a single intravenous infusion of BA259. It's a phase 1/2 open-label first-in-human dose-finding studies. The approach for the study is that we were dosing 2 subjects in an escalating dose cohort over 3 dose cohorts starting at 0.5 times 10 to the 13 gene copy per kilogram through 1E13 and then 2E13. The endpoints primarily as a dose-finding study are adverse events and serious adverse events, but also a secondary endpoint is the change in factor VIII activity from baseline. And what we've observed using a chromogenic factor VIII assay is that we do see a dose response with the cohort dosing escalation. The factor VIII levels that are achieved are sustained over time, with the longest follow-up being 18 months with no evidence of loss of expression. And none of the patients have reported spontaneous bleeds once they hit a factor VIII level of at least 15 IU per deciliter. So the preliminary results is that this seems to have a favorable safety profile, no treatment emergent serious adverse events through the longest follow-up of 18 months. There were mild to moderate elevations in liver transaminases observed in the 2 higher dose cohorts, so 1E13 and 2E13, and these patients were managed effectively with oral corticosteroids. So this is the only clinical stage gene therapy for hemophilia A that uses this AAV HU37 serotype selected for low neutralizing antibody prevalence and efficient liver-directed factor VIII gene transfer, and we'll be looking forward to the follow-up data from this study.